In this video, we'll continue on with our look at the structure of a skeletal muscle fiber. So we had a look at the basic architecture in the last one. And let's remind ourselves that we're looking at a number of myofibrils within there. And these are mostly made up of contractile proteins. Now, the question becomes, how do I end up with a multinucleated muscle fiber? Well, the reason it's multinucleated is that fibers are the result of the fusion of a whole bunch of these embryonic myoblasts which come together. So that's how I end up with the multiple nuclei. So as I said, within the cell, we'll see features like myofibrils, which are really very abundant, and they're mostly made of the contractile proteins. And we also have the sarcoplasmic reticulum, which surrounds it, and we saw that relationship in the last video. Now, what does the sarcoplasmic reticulum do? Well, its big job is to store and release calcium ions, and calcium ions are going to be very important in the action of muscle contraction and the interaction of actin and myosin filaments. We did talk in the last video about transverse tubules. They're the extensions of the sarcoplasma, or the sarcolemma, I should say, that head deep into the cells, and they surround each of the myofibrils. And they form this tunnel-like network, and they're continuous with the exterior of the cell. And they're also filled with extracellular fluid. All right. We did mention what the terminal cisternae are, but let's revisit that. Is that here we have T tubules, and we have the sarcoplasmic reticulum, and once again the widened area, the cistern, the cisternae. These are the terminal cisternae. Now, if you notice, we see the same relationship happening over and over. We see a T tubule with a terminal cisternae on each side. T tubule, terminal cisternae. So we have this grouping of 3 items, and we call this grouping, which would be once again a T tubule and the 2 adjoining terminal cisternae, we're going to call that a triad. So there's a triad, and there's another one. They're all over the place. All right. So that's the concept of a triad. Now let's talk a bit about the myofibrils. That each myofibril is made of a whole bunch of myofilaments, and the individual myofilaments come in different types of proteins. So we can have contractile proteins. They're the ones that generate the tension and cause potentially the contraction of a muscle cell. We also have the regulatory ones, which help, based on the name, help to basically determine when a fiber may contract. And then we have structural proteins that help to make sure that the myofibrils stay in the proper alignment and make sure that the fiber has the right stability, because these fibers have to be in the correct orientation for them to interact with each other and to slide by. So actin and myosin actually slide by each other when I'm generating a tension, and that's something that we're going to call the sliding filament theory. So let's have a look at the myofilaments. And we have myosin. So a grouping of myosin is what we're going to refer to as the thick filaments, and we'll have a look at a diagram. And then the thin filaments are a combination of actin along with 2 other things, tropomyosin and troponin. Now, there's also elastic filaments, and these are much like a spring. They look like a spring and they act like a spring, and this is titin. And what does it do? It helps to ensure that the thick and thin filaments can be arranged in the proper orientation. And it also, so it stabilizes the myofilament structure, and also resists extra stretching that can occur. So let's have a look. So here we're starting off with a muscle fiber. Here's a myofibril. So let's take a myofibril, have a look down here. You can see that there's sort of a constant pattern. There seems to be a pattern as we look at it. But if I look within the myofibril, it's made of a number, let's really zoom in, it's made of a number of different myofilaments. And you can see the blue ones here, and in between the blue ones you can see the red one. And these are the actin and the myosin, or the thick or thin filaments. So if I just take a section from here and have a look at it, we're going to look at what's called a sarcomere. A sarcomere is the basic contractile unit of muscle cells. And here's a sarcomere, and here's another sarcomere, and they're attached end to end. So there's one sarcomere, and another sarcomere would start here and run there, and further in the muscle it would keep going. Almost like trains on a car that are attached to each other. Sarcomeres are attached end to end. So let's have a look at one sarcomere. And each sarcomere is defined by two boundaries, this side and this side. And these are called the Z discs. So they're the two, basically the two ends. And then what we have is coming off the Z disc, we have this filament. Okay, so you can see we have some filaments coming off of there. And then you can see that in the middle we have the pinky ones, here are the red pink ones, and that's a different type of filament which we'll have, which are the thick filaments. And you can see that here's that titin, that spring-like attachment. And you notice how the titin really holds this thick filament in place. So let's have a look. What are these thick filaments? So if I pull the thick filaments out and have a closer look, you can see that they seem to have a bare area here and an area with all these heads that stick out. Now, this structure is made up of a whole lot of these, and these are the myosin that we can see. And the typical myosin has these intertwined tails, and then there's a hinge. So the myosin can actually move the position of its head. And here are the heads. Now, how do I end up go from here to here? Is that what this is basically a bunch of the myosin are basically attached. And you can see the tails on this side and the tails on this side go in different directions. So these are the various myosin tails that we see here from the two sides. And so this is a bare area where there's no heads. So the myosin heads are all here. And you can see that they go all around. They go all around the structure. All right, so let's have a look at the thin filament. So here's a thin filament. And the thin filament is made up of a number of these actin subunits. So you can see I have a string of these actin subunits, and then another string of these actin subunits, and these subunits are all attached together, and then the two strings intertwine with each other, almost like a helix. So this is the thin filament, and the thin filament is made of a number of individual actins. Now, if you look carefully, you'll see that sitting on top of the thin filament is this bluish structure, and that's tropomyosin. And if you look very carefully, actin has a binding site, and that's where the myosin heads are going to want to bind. But this tropomyosin, if you look carefully, it sits and covers up those binding sites. Now, attached onto the tropomyosin is troponin. It sits on top of it. So if I want actin and myosin to bind, somehow I'm going to have to move the tropomyosin out of the way to expose those binding sites. So that's a basic structure of a sarcomere, and this basic structure of a thick filament and a thin filament. Now, we can look at this again, that thick filaments are, once again, it's made of myosin. We have globular heads at each end. The tails are intertwining, and the tails will run oriented that way and this way. So you can see that the tails face each other from this direction and from that direction. And the heads are connected by a hinge-like neck, and each head has an active site that binds with corresponding actin. Once again, we're just looking at what we looked at before, but it's good to sort of walk through it one more time because repetition is good. And when we're looking at the thin filaments, it's actin, tropomyosin, and troponin. that make that up. And as I said, we're looking at individual actin subunits with the binding heads, and of course tropomyosin covers it up, and troponin also sits on top. And we'll have a look at how that all works. Okay, so tropomyosin, what is it? It's one of the regulatory proteins that twists around actin. As I said, it covers the active sites. And troponin is a globular regulatory protein. And this is one that holds tropomyosin in place. And I have to interact with tropomyosin if I want to move I have to interact with troponin if I want tropomyosin to move out of the way. And we're going to tell that story down the road in another video. So we're going to leave that one right there for now, and we'll see you in the next video.